So I'm very happy to be here, and thank you very much for inviting me. So I think I first met Mike a long time ago when you were visiting the Laboratoire de Physique Théorique at INS. But actually, we started to exchange just, I think, about 10 years ago when I was visiting in Princeton, and then we were very kind to invite me at Stony Brook. And then we talked about some problems in statistical inference and computational neuroscience. And I would like to tell you today about some work which are related to that, which have been done in collaboration with Aldo Batista, a former PhD student in the lab who is now a postdoc at NYU. So the problem I am interested in is how to embed multiple low-dimensional manifolds in an attractor neural network. And I want to define the problem and why we are interested in this problem. So many high-dimensional data are actually intrinsically low-dimensional, or let's say thought to be intrinsically dimensional. So this may actually be a consequence of physical laws. So for instance, if I look at VJ, okay, on my retina, I have an image of VJ, and if I move to the left, to the right, I go maybe closer or further away, I can rotate my eyes and so on, I will have different images on my retina, but they correspond to the same item, okay. So, and of course, this is also approximately true in some cases. So here you have some examples of digits 2 taken from the famous MNIST dataset, which is used by many people in computer science and machine learning. So they are just digits which are handwritten, and you see they all look like the same, and by local deformation of the two, you see you can actually relate all these two together to a single template. So there are some ways to estimate the dimension, the intrinsic dimension, which is thought to be some number larger than 10. I'm not sure I would bet so much on this number, but it's anyway something which is much smaller than the dimension of these images, which are 28 by 28 pixels, so about 800 dimensional black and white images. And the idea that high-dimensional data can actually be embedded into low-dimensional manifolds is the basis for many unsupervised manifold learning techniques, which I will not mention, but it's a very useful prior about the quality of the data and the nature of the data. So the same idea outside machine learning is also present in computational neuroscience. And so what is the idea is that, so for instance, imagine you have a population of neurons which interact together that you see here. And these neurons will have some activity. So what is the activity of a neuron? It's a set of spikes. For example, these neurons here, the red neuron. So the neuron, as a function of time here along the x-axis, you will see the neuron is active at some times, another neurons at other times, and so on. And the idea is that this population of neurons may actually express some kind of collective dynamics. So maybe in the simplest case, you could think that this collective dynamics can be written as some coordinate here, which depends on time, times some vector of the dimension, the number of neurons, right? And it's more or less active depending on the value of this coordinate. And there might be other collective coordinates embedded in this network and so on, which correspond to different cell assemblies, okay? So in this particular setting where the activity can be decomposed in a linear way, you could imagine that in the n-dimensional space of the activity of the neurons, then you would have, for instance, a two-dimensional plane here, and the coordinates on this plane would correspond to these two functions here, which are just spanned parametrically. Of course, this idea is very naive. Maybe the embedding is not linear, and you would have still a low-dimensional manifold, but a non-linear one. And the coordinates, these collective coordinates, maybe actually have a deep meaning. They can tell us something about the computation which is performed by the network, in particular how it takes decisions like motor decisions. So the question we ask from let's say a more abstract point of view is, suppose you have a neural network here, which is made of n neurons. These neurons interact together through some couplings, Jij, or they will be also called Wij, sorry about the change of notation later on. And maybe here you have a continuous variable or discrete variables which tells you about the state of activity of a neuron at time t. And then the question is, given this n-dimensional point of activity of a neuron here, how can I actually restrict the activity to lie on a d-dimensional attractor where d is much lower than n? Okay? And so what do I mean by attractor? It means that starting from an initial condition, the dynamics of this network, which actually should bring me somewhere on the attractor, and in the absence of any noise, I would stay there. It would be a fixed point. starting from another initial condition, I would go there and I would stay there. Of course, in reality, there will be some noise, so there will be some drift along this attractor here, right? And the advantage of having that is that the collective coordinates along the attractor may have a deep meaning, as I said before. So for instance, if your collective coordinate is of dimension 2, maybe it could code for the position of an animal in space. You know, I am the animal here in the space, and I'm moving in the two-dimensional plane, as I'm moving right now. So there should be some representation of this space somewhere in my brain, and this somewhere is thought to be the hippocampus. So the hippocampus is a region of the brain which is present in all mammals, and also has some evolutionary related brain areas in all vertebrates. And it has been known from the 70s that the activity of neurons in the hippocampus is related to the position of an animal in an environment. So here, I'm sorry, but the colors are actually pretty bad. It's better on my screen, but I hope you can see something. So here, you see an animal, a rodent, moving in a almost square arena here. That's the environment. And if the rodent is in one spot on this in this environment, which corresponds to this circle here, kind of circular shape, then if it crosses this disk, then there is a cell, for instance, which will fire. And this cell is called a place cell because its activity depends on the position of the animal. And the locus of the position where the cell is active is called the place field. And another cell in the hippocampus will code for another spot here, and so on. Okay, so there are about hundreds of thousands of place cells in the hippocampus of rats, and we have many more in our hippocampus. So this is a major discovery, which was, you know, one of the first bases for the notion of cognitive map or spatial maps, how we code for position and how we code for environments. And a lot of things are known about it. There have been thousands of papers about place cell and so on. I just want to say just a few points here, which in case you have never heard about them. The establishments of place fields and place cells when you arrive in a new environment depends a lot on the sensory information you can collect. For instance, visual information, it can be very important, right? But once this map is established after some learning phase, then you can actually be in the dark, move, and then you will update your position according to the map which is established, right? So you don't need only visual information it's not only a matter of processing visual information, right? It's something which is really internal. The maps are stored, so that means if you take an animal, it learns a new environment, you can put it several days later, it will actually restore the same mapping, there will be the same mapping between the identity of the cells and the position on the environment. And something which is very important is that as I'm moving here in the environment, I have a sequence of place cells which are firing depending on my position, okay? And this is related to the physical motion with timescales of the order of maybe seconds or tenths of seconds. But if I am immobile, and I'm sleeping, for instance, I can actually reactivate the same sequence of place cells at timescales which are much, much faster. And this is a crucial property, a crucial faculty, which is important, for instance, for planning, motion planning, right? On compressed timescales by a factor 20, for instance, in rodents. Okay, so that means that the activity of this place cell is supported by an internal network. It's not only something which is driven by a sensory information. Okay, so what kind of models can we build in order to try to have something which is, let's say, mathematized but not too much complicated, so highly simplified version of this reality. So in the models I will present, I will have neurons which are binary variables, so you can think of them as being silent or active. So, of course, the state of activity depends on time, so what you can think of is that time is discretized in a unit of time which is about, let's say, 10 milliseconds, 20 milliseconds, something which is related maybe to the time correlation, the correlation time of the neurons. And then whether a neuron emits a spike or not, so whether it's active or silent, corresponds to an activity state which is 1 or 0 for neuron number I. So I have if you want, binary spins. And then I imagine that through the learning phase, the sensory information I get, for instance, visual, olfactory information and so on, creates a network of couplings based on the principle of Hebbian learning, which may be, let's say, stated in a very simple way as follows. if cells fire together, they will wire together. So if cells are active together, then they have a tendency to strengthen the connection, which is a phenomenon known as plasticity. So it's a very complex phenomenon. Here, what you get is only a very highly simplified version of it. It's just a caricature. But anyway, so imagine you have two neurons. It should be a green and an orange one. If the centers of their place fields Rij, sorry, the distance between the centers of the place fields is such that the two neurons do not, the two place fields do not overlap. They are essentially never active together. So they cannot strengthen the connection, so they will not be connected, okay, in the network. If they overlap, that means that there are a lot of positions when the animal goes through, they will fire together. So I will build here a coefficient, Jij, in between. So at the end of a learning process, I'm assuming that Jij, the coefficient of interaction between neuron I and J, depends on the distance between their place fields in physical space. And it's something like a short range interaction here, and it goes to 0 at long distances. Okay? And long means of the order of the diameter of a place field. Okay. So now imagine you have many neurons, then, well, I don't know if you can see something actually. I can see on my screen, but not here. So you can see there are many place fields with many neurons, yes? So you see that if two place fields overlap, I will have a connection. So I get a map here, a network of interaction, which actually represents the topology here of the place fields, right? So of course, this picture here you should be careful here. The position here is well defined in physical space, but the corresponding neurons have position in the hippocampus, which have nothing to do, right? So actually, the true thing is something which is much closer to that. There is no connection between the fact that two neurons are close to each other in the brain area and that they code for place fields which are close to each other in reality, in physical space, right? So this is why when people do experiments, they put an electrode somewhere in the hippocampus, they measure the activity, and they pick up neurons which are close to the tip of the electrode, and they can code for positions which are completely far away in real space, right? Okay, so now, if we imagine such a network of interaction, such a graph of interaction, what is going to be the distribution of neural activity configuration? So I will do something which is inspired by equilibrium statistical mechanics, which is obviously a high simplification, a very strong simplification of reality. And so imagine you have your set of binary variables. Then you can imagine a Markov chain here, where, for instance, I can go from one state of activity to another one by flipping spin number I from 0 to 1 with some rate. So probability per unit of time R, and you can do the reverse thing here with rate 1 over R. And the log of the rate is typically depending on the activity configuration in this way. Each neuron, and this is based on the neurophysiology here of neurons. So a neuron here is summing up all the activity of the presynaptic neurons with some weights which correspond to this interaction here. And if the sum is above some threshold, then it will have a high probability to fire. If it's below the threshold, it will have a low probability to fire and will be silent, right? So here, this is taken into account in a simplified version by this activity, by this rate. I hope this is clear. And yes, more or less. Okay, so essentially, once I have defined the dynamics over my set of spins, I'm just having a dynamics over the n-dimensional hypercube. And you know that since this dynamics is actually satisfying detailed balance, eventually at long times it will be the probability of any configuration of the n activity state of the neurons will be given by this exponential of this term here. And so this is just the Gibbs equilibrium measure of a kind of Ising model where the interaction between neuron I and J depends on the distance between the centers of the place fields. Okay? And you have a term here, the threshold gives rise to a term which corresponds to a magnetic field. So you have positive interaction here, so which favor a ferromagnetic phase. They are short range if you want, and you have here a negative field which is trying to push down the whole activity. Okay, so in fact this model was introduced a long time ago in the context of lattice gas model in order to study the liquid vapor transition, in particular by Lebowitz and Penrose in the 60s. So here what were the neurons? So these people were not talking about neurons, they were talking about occupation numbers. Imagine you have, for instance, a two-dimensional lattice, but you can do that in 3 dimensions, obviously. And you have some occupation numbers, which are 1 if you have a particle on this side, or 0 if you have no particle. And if you have some kind of Lennard-Jones interaction potential, so first of all, you have very short range, very strong repulsion, so you can put at most 1 particle on each side. If they are next to each other, then this is the bottom of the well of a Lennard-Jones potential, so this is attractive. So you get a positive interaction Gij between the 2 neighboring sites. And if they are far away from each other, no interaction at all. And the magnetic field here is just controlling, it's just a chemical potential which is controlling the total number of particles on your lattice, right? And now it's well known that if you simulate configuration from this equilibrium measure, what you will see is that at high temperature is a uniform density of particles in space, and at low temperature you will go below the ferromagnetic-paramagnetic transition, so you will have a bump of very high density, which corresponds to a droplet of liquid, which is surrounded by very low density, which is the vapor phase, right? And obviously, the interesting thing is that since this Hamiltonian here, the interactions are invariant under translation, that means that I can actually move my bump anywhere here in the space, and I get exactly the same energy, right? So I have built here from an n-dimensional configuration of occupation numbers, a 2-dimensional attractor, okay? Because I get this bump naturally, which will form, and then by a small force, I can move it very gently anywhere in space, and it will stay in the same shape, modulo thermal fluctuations.
Sorry, Rémi.
Yes?
Maybe I missed it, but the number of neurons firing is fixed.
So here it's fixed. You can do this exactly because here usually you have a fixed number of particles. So you say, for instance, 10% of your sites are occupied. So what is going to happen at low temperature is that since particles, they try to aggregate, to cluster, you will get this big cluster. And of course, at finite temperature, there will be a very low density vapor around it, right? And you can move this droplet of liquid at no cost. So it will diffuse. So we have built here, you see, an explicit way of building a 2-dimensional attractor or 3-dimensional attractor from an n-dimensional problem. So now the interesting point, so this has been known for a long time. So the interesting point is what happens if you want to embed more than one continuous attractor in your recurrent network, right? In your statistical physics model. That becomes much more interesting. So why do we want to do that? Because it's known, obviously, as animals, we know that we can memorize many environments. And in fact, it has been shown also in rodents, in experiment, in this very nice paper here. 11 maps for 11 rooms, where the same animal was put in 11 rooms, and I think people stopped at 11 because they got bored. That was very boring to do more than that. And you could record the activity in the hippocampus, and you could see that this animal, this rodent, had different representation, 11 different representation for 11 different rooms. Actually, the story can be more complicated than that. Some animals may have more representation for the same room. But OK, we don't want to go so far. But still, I mean, there is a single hippocampus, right? So how is it possible that there are 11 different continuous attractors storing the position of the animal? And as the animal is moving, this position is updated along the manifold here in a single network. So essentially, what I want to do is I want to move from these simple things where I had a single high-dimensional space. I have it somewhere. Yes, with one attractor into something which is fraction, into basins of attraction with many, many attractors. And the question I would like to ask is how many attractors I can actually store or I can embed in a network with n neurons? This is the question I would like to ask. Okay, and in fact, we would like also to say something about this number, and more precisely, how it depends on the quality of the attractor. Because you see, you can do two things. Maybe you want to store many maps in your networks, so the number matters, but the quality of the maps is low, actually. You have a bad spatial resolution. Or maybe you don't care, you really want only a few maps, but you want very precise maps. So what is the trade-off between the quality and the quantity of maps? That was the question we were interested in. So why is it important also so it's important to be able to memorize different environments when we move from one environment to another one. But sometimes it's important to have different representation of the same environment depending on the context. And there are beautiful experiments showing that, and I want just, this is a cartoon, not taken from the experiment, but it gives the idea. If you take an animal, and this is its natural environment, for instance, then in the presence of food, then maybe what is going to happen, that a lot of place cells will code for interesting position in this space, so you will find a lot of place fields around, condensed around the position where the food is, because it's important for the animal to know precisely where this food is. But suddenly, if the context is changing and you have a predator, what matters is not food, but the shelter. So, and the shift from one representation to the other one should take place very quickly. So that means that in this space here, then depending on the contextual information, then one should be able to move very quickly the activity configuration from one attractor to another one. And this will have consequences on the behavior of the animal. Okay, so how can we embed more than one attractor? So first idea was proposed to do that by Samsonovich and McNaughton in 1997. And actually their idea was directly inspired from the Hopfield model of statistical mechanics from the 80s. And the idea is the following. Oh, okay, so what you can say is that suppose you can, suppose you take neurons I and J. In the first environment, so for instance, the orange and the blue one, in the first environment, maybe the place fields are far away from each other. So if you were to draw a network of interaction for this environment only, there would be no connection here. But maybe in another environment, they are next to each other and you would draw a connection, right? And so on. So what you do is that you do exactly what I was doing before for this lattice gas. You have a matrix of connection for the first environment, which depends on the distance between the position of the place fields of these two cells in the first environment. And you do that for the pair of cell ij. Then in the second environment, they might be further from each other or closer from each other, so you get another connection and so on. And then you assume as the animal is learning all this environment, then you just add on top of each other all this contribution. Obviously, this is highly simplified, because maybe older environments can be a little bit forgotten and so on, or they are to be reactivated. But we forget about that. Imagine you just sum up all these contributions, right? So now you have a matrix which has a meaning in each environment. So if you have but all the connections are mixed up, right? If you look at your network of interaction here, then you cannot see from which environment these connections are coming from, right? So the question is, if you simulate the model corresponding to the summation of all the different environments, what is going to happen? So let me show, for instance, a simulation where we have 8,000 neurons here, 90 by 90, and we embed 3 different maps in 2 dimensions. And so what I'm doing, I'm just considering the equilibrium distribution with this Jij matrix. So I'm just running on a very simple Metropolis Monte Carlo algorithm with the condition that the activity is fixed. So you don't flip one spin at a time, but you flip 2 spins in order that the activity is always conserved. And so I have a vector of activity which is made of 0 and 1, and I have 8,000 of them. And I will plot this vector of activity in 3 different ways. So it's the same activity, but plotted here in 3 different ways. So in the green way here, I will, if a neuron is active at any given time, I will draw a green dot at the center of its place field in this environment. And the same neuron will be active somewhere else here and somewhere else here in the blue and the red environment. And if it's silent, then it's obviously silent everywhere. I don't show it. Okay? So it's the same activity configuration shown in 3 different ways. So here are the Monte Carlo simulation. So we start from a simulation with an initial condition where the bump of activity is actually in the red environment. So that means this activity is coding for position in the red environments. That means the animal is somewhere at this position, 2-dimensional position in the red map. And then just by thermal fluctuation, you will see jumps to other attractors, right? Because they are all there embedded in the same coupling matrix. So if you wait a little bit, you will see that the bump is going to materialize in the blue environment. Then we see it now. And then it will jump back, and at the end, when the simulation stops, I think it stays in the blue one. Okay? And if you just wait enough, you will see that it jumps from the blue, the green, and so on from time to time, right? Because they are all possible attractors of the dynamics. And they correspond to different equilibrium states. So we have three continuous set of continuous states here, equilibrium state. Okay, so the main theoretical result coming from this is that, and I won't show the calculation, but there is a phase diagram in the plane of, so two things here, temperature and the ratio between the number of manifolds you want to embed divided by the number of neurons. So of course, if temperature is too high, then you get in the paramagnetic phase, and no, there is no information, like in the, so it's always as being in the vapor phase, right, in the gas phase. You want to be in the liquid phase in order to get the bump of activity. So this phase is trivial here. The one here is trivial also because there are too many manifolds that you want to embed in your GIJ matrix, and what you create is a spin glass because they all try to push the activity of the neurons in different direction if you want. So that's very bad, so you cannot create anything. And if temperature is low enough and alpha, this ratio L over N is low enough, then you are in a phase where you can actually create a bump of activity, localized, in any of the environment. So it's very nice because it tells you that you can embed a number of environments which is actually extensive in the number of neurons, with a factor which depends on the dimensionality of the manifold, which can be computed. So it seems very nice, but it's not so very nice, because the problem is that the fact that you can create a bump and have a bump somewhere in the environment tells you nothing about where you can actually localize it. And the point is that it's a necessary condition to have a localized activity, but it's not sufficient to be able to code for all the position in the environment. You want actually to be able to put it everywhere, right? In order to be able to code for any position. For instance, if I want to code for a two-dimensional map. And the fact, the truth is that this is not going so well here. This is not what is going on, because if I look at my kind of effective landscape as a function of the center of a bump in one environment, then this is not flat at all. This is not invariant under translation as this used to be in the case of a single environment due to the invariance by translation of the interaction, simply because the other maps I am trying to encode in the other submatrices are creating some noise. So my landscape here I get is not flat at all, and it will create just by chance some wells here where my bump will be trapped here. Can be trapped here, and you see the distance between these two positions here can be large. So I get a very bad spatial resolution. So this is not telling anything about this kind of resolution, spatial resolution capacity trade-off I was mentioning before. So what we propose with Aldo is the following thing. So suppose I want now to build again a memory for a certain number of manifolds. I would like to impose a spatial resolution. I would like to compute the critical capacity, which is the maximal number of manifolds I can embed as a function of a spatial scale here, epsilon. So what I will do is that I will forget about continuous manifold. Anyway, this anthologic McNaughton prescription is not producing continuous manifold. I will have discretized maps. And how do I do that? I will impose that, for instance, on this blue map, I will just draw randomly some points. And the distance between nearest neighbors is epsilon, which I control. Okay? And I want all these points here to be fixed point of the activity of the dynamics of the neurons. I will show you the dynamics in a moment. So I'm sure that actually there will be a lot of fixed points here, so I can put my bump here, there, and so on. And since the distance between two neighboring points is epsilon, I'm sure that the spatial resolution will be at most epsilon, right? My error will be at most epsilon. So by increasing the number of points, I will have a better epsilon, and I will have a very fine map. Of course, if I do that, maybe the number of manifolds I can embed will be will be lower, right? So the question now is how is scaling the capacity as a function of epsilon? So let me define more precisely the model. So I have n neurons with indices I, L manifolds, which will be just hypercubes, so very simple manifolds, no complicated structure, with indices mu running from 1 to L, and they are all d-dimensional manifold. So in d equal 2, you can think of L, you have L squares here. So for instance, here you see Again, this is very bad. This looks much better on my screen, but okay. So you see here, you have an example with 5 neurons, 2 manifolds in dimension 2, there are squares, and I choose a number of points. So my epsilon, instead of epsilon, I will use p, which is the number of red crosses, the number of points I throw in my manifold, here on my manifold. So here you see 3 points. That corresponds to 3 positions of the animals. You see 3 orange points here, 3 violet points here. Okay, and how do I build configurations, binary configurations on that? Once I have this position here, I look at all the place fields of my neurons, which are drawn randomly. Each time the point here is inside the place field of one neuron, I would say that the corresponding neuron is active, it's 1. If it's outside, this is 0. So for each point, I will draw a sequence of activity of the different neurons, the n neurons, which is a binary vector of activity. Okay? And that codes for the activity configuration I expect if the animal is there in this particular environment. Okay, so now how is p the number of points connected to the scale epsilon? That's very simple. So for instance, if your environment is of side 1 here, linear length scale is 1, then you know that if you draw p points randomly here, you know that epsilon, the distance between numbering points, will be of the order of p to the minus 1 over d, right? So by increasing p, I will decrease the resolution scale epsilon. Okay, it's the same thing to control epsilon or p. So this is how I do exactly, and I draw randomly the centers of the place fields. The diameter is fixed. I draw randomly the p points in each manifold, so I create a certain number of configuration here, and then I would like to store this configuration in my network. Okay, so what is the dynamics? The dynamics is very similar to the one I've shown before. Given the state of activity at time t here, where t is a discrete time, I will simply multiply sigma by w, which is the interaction matrix. Sorry, it was called J before. I'm sorry about the change of notation. And then I apply a non-linearity. If the argument is positive, then the neuron I will be active at time t plus 1. If it's negative, then it will be silent at time t plus 1. Okay? So it's just a thresholding of the things. And what I would like is to find a matrix W such that all my activity configuration here satisfy this equation. Okay, so it should be fixed point. So if I start from one of these activity configuration, I apply this dynamical rule, and I should end up exactly in the same activity configuration. So this is a minimal requirement, but actually I will ask for a bit more than that. I will ask for, if I want sigma I to be 1, I want the argument to be as positive as possible. And if sigma I is 0, I want the argument to be as negative as possible. So that's good. It makes the network more robust if there is a little bit of noise, right? So how do we write now the mathematical condition? So for each environment mu, for each point m in the environment mu, that is the activity configuration for neuron J. So the activity state of neuron J here, which I've drawn. I want this term here to be, so if I want sigma I to be 1, so it should be, I will consider this term here. If I want sigma I to be 0, I will consider minus this term. And then I look at the worst case, which is the worst point, the worst environment, and the worst neuron. And worst means that this term here is minimal. And I want this minimum here to be maximal. So I want to choose the W so that this minimum over the neuron, the environment, and the point is as large as possible. So it's a max-min condition. So I want the worst situation to be as good as possible. So I will push W in order to maximize this minimum thing, right? And in fact, it's a condition which is encountered in machine learning, in support vector machine, all machine learning, nothing deep here, okay? And so it means that you want to make the decision, the boundary here as far as possible from your point. Okay, and there are algorithms to do that, right? Support vector machines have been studying for such a long time, so there are very efficient algorithms to do that numerically. And if you do that, I'm showing you just an example. So the red crosses here are the fixed point, are the points which are drawn randomly in the space. You see here the neurons which activate or deactivate depending on time with periodic boundary condition. And you see what you get is a coherent bump. There is a single environment in that case. so due to periodic boundary conditions, so you see it goes away from through the right, and then it goes back to the left, or the up or down. So you see essentially one collective bump which is moving on the noise and diffusing in your space. Okay, so that works. That shows that if you have a sufficiently small epsilon, which is the typical distance between red crosses, you get a kind of continuous motion. I mean, obviously this is not continuous, but it's not so bad. Now let's do that with two environments. So again, I start with the initial condition where I have a bump in the first environment, then it's completely random and uniform in the second one. And as you run the simulation, you will see from time to time some jumps from one environment to the other, right? So it's very similar to the Samson-Magic-McNaughton phenomenology, except that here the scale, the spatial resolution is totally controlled. This is epsilon, as we choose by the number of points. You should see the jump at some point. So it's always in the first one, but then at some yes, you see now it's on the right one. You see the bump, right? And you can do that with a certain number of environments. And my question is always the same: how many environments can I? In how many environments can I do that if I fix the number of red crosses? Okay, so how do we compute that? So I will use techniques which come from the statistical physics of these other systems, which were invented after the discovery of, the use of replica techniques on spin glasses and the discovery of replica symmetry breaking by Parisi in the 80s, and in particular in a framework which was invented by Elizabeth Garner at the end of the 80s. So what we do is simple. Is simply we look at all these conditions. So I want all these inequalities to be satisfied. Or it's even better if I can put, instead of 0, I have a kappa here, but let's put 0 just to make life simpler. And if I have a small number of points or environments, then in my space of all possible coupling matrices, you can imagine you have a volume of solution. So here, this is the feasible set of solution, this blue one here that you see here. And if I increase the number of conditions by asking for more points or asking for more environments, I will decrease this volume, right? And maybe I will end up in the yellow. If I increase it again, I will have a red one. At some point, it will go to a single point. And if I am above this number of conditions, then I'm done. That means I cannot store anymore, and this is the critical capacity. So essentially what we want to compute is the log of this volume in the coupling matrix space as a function of a ratio L over N, and that should depend on the number of points P, and I will see where it goes to, the log is going to minus infinity, and that will define the critical capacity. So the calculation is non-trivial here compared to what was done by Garner. So Garner invented this formalism, so it's a beautiful one, but I mean, she had independent patterns, so independent vectors here. Well, they were just drawn randomly. Here I'm drawing them randomly, but they are correlated because they are just emanating from this low-dimensional manifold, right? So that creates a lot of correlation between the components of the configuration. So we can do this calculation. It involves p-dimensional integrals. So up to p equal 10, we can do a calculation exactly. But then for large p, which is what we are interested in because we want small epsilon, we cannot do that exactly, but we have, I think, rather solid arguments to say that we expect the measure over the weights W to become asymptotically Gaussian for large p. So what we do is first a double infinite limit. L is sent to infinity, N is sent to infinity at fixed ratio, and then we send p to infinity. Then in this limit, I think things become Gaussian, so we can actually solve the equation. And I want to show you two things. First, the results, and then a little bit about the connection with random matrix theory. So here you see P in the x-axis and the critical ratio L over N on the right axis. So for P equal 1, that means that on each manifold, you can guess there is a manifold here, you have a single pattern. So the fact that it's a finite dimensional manifold plays no role because you have a single one. So what you get is alpha C of P equal 1, which was found by Garner in 1988. And if you put more points, for instance, 2 points here, then you expect that the capacity is going to decrease because you are putting more stuff in your manifold, right? And it's decreased more or less as alpha C1 over P because that's the number of condition inequalities you have to satisfy. But actually, as you increase P, you see that it decreases less quickly than alpha C1 over P. simply because these conditions are not independent from each other. They are not orthogonal to each other. So they are a little bit redundant. And the more stuff you put on your manifold, the more redundant they are. And the more easy it is to satisfy a new one if you have already satisfied the one before, right? So it decreases at a rate which is much lower than 1 over p. And the question is, what is the asymptotic scaling with p? You see, for instance, if you have 10, you are here, and you would like to know what is going on when p goes to infinity. So I won't do a calculation because it would take time, but I just want to say that there is a deep connection with the random matrix problem, actually a Euclidean random matrix problem. So what are Euclidean random matrices? I'm sure that some of you already know, but so they were invented some years ago by Mézard, Parisianzi, or at least from the point of view of physics. Maybe they were already introduced in math, but I'm not sure. And so the idea is the following. So in random matrix theory, there are many statistical ensembles. So the simplest one maybe are when you have a CAG. So this is my matrix, the entries of a matrix. As you can imagine, for instance, they are drawn randomly, independently from each other. Okay, that's a very simple example. But you can imagine that you have another distribution where what you do, you have a square here or a cube, a d-dimensional object. You draw randomly n points. For each pair of points ij, you compute the distance. And then Cij will be a function, deterministic function of a distance. Since your points are random, the coefficient will be random, but they're all correlated. Because from n times d coordinate, you compute n squared coefficient. Actually, n squared over 2, because it's a symmetric matrix, right? Okay, so, and this is an ensemble which has been studied a lot. So what we consider here is a kind of non-trivial, trivial limit of this ensemble. So what do I mean? I mean, if you take a single space here, and you keep it, so the length here is, let's say, 1 fixed, and you put more and more points, then actually this statistical ensemble becomes trivial. Because if they are just uniformly put here, essentially what you get is that the top eigenvalues, or the top eigenmodes, sorry, are Fourier plane waves, because you are just sampling uniformly your space, right? So you get Fourier modes. Okay, so this is trivial, but what is non-trivial in our case is that we have many spaces, right? And that's a situation which was not considered in physics, because usually when you put particles in space in physics, you have a single space. But here we have a single set of neurons, but they code for many different spaces. So the same neuron I and j can be at some distance in the first map, at another distance in the second one, at another distance in the third one. So we just sum up all these Euclidean random matrices. And then you get a big matrix, and now it's non-trivial to find the spectrum of this matrix. And if we have it, then we can derive the capacity. So it's possible to find it with standard techniques for random matrices. And then just to go to the results, we find that the asymptotic behavior of a capacity is as 1 over log p to the d, where d is the dimension of a manifold. So what does that mean in practice? That means it decreases very slowly with p and very slowly with epsilon, the scale resolution, the spatial resolution, which is p to the minus 1 over d. So just to give you a concrete illustration, suppose you want to store maps with spatial resolution epsilon. You will have a certain capacity, a certain maximal number of them that you can store. So epsilon is compared to your size is maybe 10 to the minus 6. It's very precise. But you say, oh, no, I don't want 10 to the minus 6. I want 10 to the minus 12. So I want to go from epsilon to epsilon squared. So the math will be much, much more accurate, right? So what is the number of maps that you can store now? It will be much lower than before. And what you see is that by going quadratically in the scale resolution, you actually go only, you lose only a fraction of your maps. So the scaling is actually very good, right? So you can increase a lot the resolution, and you lose only a fraction of what you had before, right? So it's a good situation. Okay, so I think I should be more or less done now. I have 2 minutes. So I just want to end up with a conclusion. So what I've shown is I've just recalled the model for single continuous attractor, which was actually a model which was invented in the 60s, so it's a very old model. So it was invented also in the context of computational neuroscience by Sushi, and there are beautiful experiments, not for place cells, but for head direction cells, for flies, where actually there is a direct proof of the presence of this cell. So it's a beautiful thing. So now so you see that's an example of biology where theory was done in 1977, and experiments were done, I don't know, just 5 or 6 years ago. Yeah.
2018.
Yes, yes, okay. So you see it's predicted 40 years before. So that's a beautiful example. Okay, so for now the question of, which is much more complex, of multiple continuous attractors. So the additive learning rule, which was proposed 20 years ago, shows an extensive capacity but totally uncontrolled quality of the map. So I think it should be essentially ruled out. The optimal stability rule I'll propose gives quasi-continuous attractors. So it's nice. We can change it in many ways. We can put some constraint on sparsity of W. Not all pairs of neurons interact together in real neural network, of course. So we lose, obviously, in terms of capacity. More complex geometries, non-homogeneous distribution of anchoring points, and so on. But I think the most important thing is that the rules we use actually to define this optimal stability network of interaction, they are not biologically plausible. So maybe they give us an upper bound of what is possible. But now, if we have a biologically plausible learning rule, we should be able to actually reach only a fraction of that, and we don't know how much. And so I just want to mention, and I will finish with that it's a very interesting question. And there might be actually nice experiments, recent experiments, which give us a little bit of insight about what is going on. So it's a beautiful paper by Farouk and Dagoy, which was published 2 years ago, 3 years ago. So what these people do, they look at very young rats. So just after the birth, and it's known that around day 13 after birth, the place cells emerge. And essentially that takes place more or less at the first time the rats are able to open their eyes, right? Their eyelids. So as soon as visual information is coming into the animal, then you see place cells. And how do you see place cells? So here is the experiments. So you take the animal, and the animal is actually running on a corridor here, which is 1 meter long. And what you can do is actually, through electrodes, measure the activity of place cells in the hippocampus. And you see that if you order properly the activity, the mean activity as a function of the location of the animal when it's running, you see more or less a sequence of place cells. So some of them are very nice. They have an activity which is very much localized. Some of them are not as nice. You see there are regions where it's not active, but it's more localized at the end, but you see it's not perfect, not as nice as in adults, but more or less place cells, right? And this is when, so you can identify the position for which the cell is coding when the animal is running. And now is coming the very fun part of the experiment. What is happening when the animal is sleeping? So when the animal is sleeping, then what you can do is record the activity, and then using the inverse mapping here, decode the position corresponding to the activity, right? So since the activity is changing over time during the sleep phase, then you have a kind of virtual trajectory in space, right? And people have done that in adults, and it's very nice. So for instance, if adults have adult rats, if they have learned an empty environment, you see essentially Brownian diffusion. If there is a spot where there is water or reward food, something, what you see is something which is really well fitted by on-style Uhlenbeck process, where you know some kind of spring going back to this position. So now what is going on if you do that in puppies? Well, you see something which is very nice. So let's look, for instance, at so there are many data here in this picture, but I mean, let's look, for instance, at this thing here, only at the top row here. So there are just repetition on the same experiment. So let's look at the top panel here. So here, this is when the animal is sleeping at day 15 or 16. Essentially, you get the same thing. And you see that, so here you see the position, which is decoded as a function of time. So again, you measure the activity of the neurons, you decode the position, and then you plot it as a function of time. And you see the virtual trajectory is not moving. The position is stuck there, which is the end of the corridor. This is at day 15, 16. So place cells have been existing for 2 days, but you know it's completely stuck. Day 17, 18, then it's moved a little bit. 19, 20, it's moved a little bit more. And if you go to the end, 23, 24, people were not able to do the experiment at longer time. I think because actually the head size is growing so fast that actually the surgical implant is not maintained, I think. You see, you get to more and more continuous behavior. So I would like to see that as some kind of evidence for the maturation of a continuous attractor as a collection of discrete attractors which are actually merging, right? So it's just a way to interpret the things, but it seems pretty clear that we are going from discrete fixed point to something which is much more continuous over time. So I think these are beautiful experiments. And maybe we see that epsilon is, I would like to see it as epsilon decreasing with time, but it's a little bit too much. But I mean, there is plenty of motivation to try to study this dynamical process of learning. I think I should stop here.
Questions?
So, can you say a little more about the experiments, Rémi? So, how close to reality is this idea of tuning up the couplings Jij between neurons? I mean, is this really what happens in the brain?
So, honestly, there is no direct proof for this process in living animals. Okay, so I don't know. I mean, so what is known is that plasticity mechanisms, plasticity effects exist. They have been measured in vitro. There are also some experiments in vivo, but not as much control. And there are many mechanisms. So people have discovered a phenomenon which is called a spike timing dependent plasticity. So you take two neurons. You know that there is a presynaptic neuron which is connected to a postsynaptic one. You stimulate this neuron at a time, and then you stimulate the second one with a delay delta. Okay, and you repeat this process, always with the same delay. And then what you can do, so first of all, before doing that, you stimulate the presynaptic neuron, and you look at whether you get a spike, you get an activity, a response in the postsynaptic one. And that you would get, right, or not. So maybe you can compute the probability that you get with. So that gives you an idea of the strength of the coupling. Then you do the stimulation protocol, right, many times. Then you stop, and then you stimulate here, and you see what is the probability of getting a spike in the postsynaptic neuron, and you see whether it's smaller or higher than before. And what people see is that they can plot as a function of a delay between the presynaptic and the postsynaptic. They can see how much the synaptic coefficient, or let's say the proxy for the synaptic coefficient measured through this increase of or decrease of, is going as a function of delta, and what you see is something like that. You see when the delay is large, you see no change. When it's small and positive, you see a big change. And when it's small and negative, you see a big depletion of the synapse, right? So that means that, and this scale here is, I don't remember exactly, but it's the order of, I think, 40 milliseconds. And then the effect is gone, right? So that means that if you really impose some activity like this, right, in order to satisfy causality in some way, then you have to increase the coefficient. So it will stimulate the interaction. If you impose something which is reversed, then that depletes the synapse in order to go to this activity kind of relation, right? So now what is going on in living animals in the hippocampus? We don't know. There are very few results about the physiological connection in CA3, which is the region of the hippocampus of interest. There was a paper in Science, I think 2018. The connectivity is very sparse. There is no proof that the same mechanism is going on. It's a reasonable assumption, but I don't know. Honestly, I don't know. I mean, obviously, this Hebbian mechanism, fire together, wire together, it's very simplistic, right? It's but you know, there should be something taking place similar to that to some extent. Yeah.
So as you know now, there are a bunch of experiments showing that that place fields actually code not place but more general contexts for behavior.
Yes.
And in particular can respond not just to space but to smells, sounds, and also to abstract things like
yes.
you know, the lengths of necks of artificial objects.
Even the task that the animal has to perform.
Etc. So do you have any thoughts about how in such representation of abstract spaces this kind of thing would work?
That's a very good question. So if we want this thing to apply, I'm not sure we should, but if we want, right, then that means we should define along this orthogonal direction coding for smell, coding for emotional state, coding for task, for behavior, and so many things, right? We should invent some kind of positions and then some kind of metrics along this direction, which is obviously, I don't know what we should do, right? But in principle, we can have higher dimensional space, but certainly the d dimension of position in space should be physical space should be very different from the other ones, right? So that I would like to think about that, but I don't know how to do that without making so much assumptions, you know, which are not justified by in any way. But I fully agree that what we see here, I mean, when people do when they think about place cells and they measure the position and they measure the activity of an animal as a function of its position, it's only a very low dimensional projection of something which is much higher dimensional. And there are many other axes. I fully agree. Thought about that, but I don't have any good way to do it, you know, without very heavy assumption about what it should be and what that should be the distance along this direction and so on. I don't know.
I have a question. May it be so that when we give some drugs to the animal, the parameters of the system change? For example, temperature increases, at some point phase transition occurs, and the animal loses spatial awareness. Does this process look like a phase transition?
So what people have seen, for instance, there are some experiments where you can see that depending, so what they do, for instance, they have an animal moving in a maze, and you can see the presence of place cells. Then what the animal has to do is for 10 trials in a row, it has to go to somewhere in the maze. And then the next 10 trials, it has to go to a different position. And the animal is rewarded to do that. So after some time, the animal learns to do that. So it knows from the 10th to the 11th, it has to change, right? The rule has changed, it knows it. And he does it with a very high success rate. So what you see is that some place cells keep the same place fields, you know, in the first 10 trials and the next 10 trials. They code, for instance, for, you know, the beginning of the maze or whatever. But other ones, they remap and their place field change completely from the trial number 10 to the trial number 11. So they clearly dependent of the on the task the animal is doing. So we need a mechanism for that, right? So here, this manifold, they are just discrete and far away from each other. So it's a very highly simplified thing, right? But I like the idea of trying to understand the trade-off between capacity and resolution, but reality is much more complex than that. I fully agree.
These place cells recording which you showed, so we see that there's some representation of space in the way they activate it, and then so when a posteriori there's some reliving of the experience probably when they active in the same temporal sequence. Is there any in this experiment, is there any room for seeing the prediction? So do we predict when we move that we will go a certain way?
Yes, there were.
Something you explained in the beginning that there is different time scale which we use.
Yeah.
But can you resolve it in this situation?
Oh yeah, so there are beautiful experiments where people, for instance, you have an experiment I think that was with Buzzaki, I think in Buzzaki, where there was an animal. So the animal had to move in an environment which was like this. So these two things here are, you know, regions where the animal cannot go, and when the animal is actually has a wheel here, you know, so it can enter the wheel and just walk on the wheel and so on. And then once the animal exits from the wheel, it can go left and come back, or it can go right and come back, right? And what you can see is that, so when the animal is moving, you can record the place cells, and you can add, so understand the mapping between the place and the activity and the identity of cells. And when the animal is there, you can see sequences, okay, of firing, which correspond to the left or the right trajectory. Well, it's not as perfect as that, but I mean, definitely there is some signal. And then this is correlated to what the animal is doing next.
Later.
Yes, so it's clearly planning.
There's a prediction. Okay.
Yes, yes.
It's imagining.
Yes, as you want to call it. But clearly, I mean, it's planning some kind of future physical motion, yes.
But only after certain prior actual trials of doing this.
Yes, I mean, it has to know the environment first. I mean, there are some issues about pre-play. I mean, we are getting into complicated things, but yes, it has to learn it.
I mean, you could teach it in one environment, one maze, and then subject to a completely different one. So, and then see whether before going into a new maze, it will replay.
Yes, so there are experiments where the animal knows different environments, and you can see in one environment some replay of the other one. Yes, that you can see. There is an experiment by the mother and Jacek, and where they see that There are also experiments by Karim Benchenan at ESPC in Paris, where actually you do this kind of things, where it's a Y-shaped maze, if I remember correctly. The animal has to learn that it has to go left, but before it understands that it takes some time, and it goes sometimes right. So you record the place cells here, and then after some time the animal is always going left. Then the animal sleeps, and when you do during the sleep is that you stimulate these place cells. And when it wakes up, sometimes it goes right. So you have changed the memory. Yes.
Can I add a comment?
Yes.
So there's an alternate hypothesis that people are talking about these days, besides the idea that you can code place of the hippocampus as a predictive map, not as a map per se, but predictive map. And so there's this very interesting formulation that people have in the context of machine learning, actually, as they imagine when you're learning to navigate spaces, you need a module that can predict where you go, and then so that you can do planning. So people are starting to talk about places not as the actual cognitive, the where you are, but where you want it. Questions? Let's thank Rémi.
Thank you.